Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
So as you know, I don't know whether you know this, that I think it's between about greater than 2% of all of the energy that's used globally is used to fix nitrogen. That means to make nitrogen from the atmosphere or from fossil fuels. So it's a hugely energy-intensive process that makes fertilizers. What happens with most trees, nuts, and wine is they have what you call a juvenile phase. So a juvenile phase is when they are growing vegetatively. That means they're producing, just vegetative material, roots and leaves and branches and so on. It's a really good process where they have to train the plant to have 2 codons that are then sit on a scaffold, and then they have these shoots that come out, and then they train the shoots so that they will get into production faster. One disease, crown gall, that I've been focused on for the last many years, trying to find resistance to crown gall disease. This is a tumorous growth that many fruit trees and wines are very sensitive to, and we have come up with a solution.
Forget frequently asked questions,
common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Abhaya Dandekar. He's a distinguished professor at UC Davis. He's a director as well, part of the Plant Transformation Facility. We're going to talk about fruit and nut trees and the traits in them that I guess help them succeed or not, phenotypic traits. So, Abhaya, thanks for coming. How are you doing?
I'm doing great. Thank you so much for inviting me to participate in this podcast. Yeah.
Oh, you're welcome. Tell me a bit about your background. How did you get into studying fruit trees?
Oh, I started my career as a microbiologist studying microbes, and mainly those that fix atmospheric nitrogen in plants. As you know, nitrogen is a key element that's required for all living forms, whether they are plants or microbes or humans. And I started my life working with this bacteria that fixes atmospheric nitrogen, and it really drew me into the whole world of molecular genetics and how genes move and are expressed and provide really useful traits. The ability to acquire nitrogen from the atmosphere is a hugely important trait. But that's what I started my research on, and I work now with trees, nuts, and wines.
Okay. Well, before we get into the trees, the nuts, and the wine, can we talk a little bit about nitrogen fixation? Would that be okay?
Yeah, I'd be happy to, yeah.
Bacteria that would associate with certain plants fix nitrogen out of the air, or how does the whole cycle work?
Yeah, so as you know, I don't know whether you know this, that I think it's between about greater than 2% of all of the energy that's used globally is used to fix nitrogen. That means to make nitrogen from the atmosphere or from fossil fuels. So it's a hugely energy-intensive process that makes fertilizers. And as you know, with the Ukrainian war, Ukraine is one of the big suppliers of fertilizers worldwide. So you need fertilizers to grow plants or grow crops because there's not that sufficient amount of fossil nitrogen in the soil to allow productive growth of agriculture. And so you require nitrogen. The reason why they burn down forests because, they're taking advantage of the fixed nitrogen that's there, in these old forests that they can use to grow crops. So nitrogen is really a key element that is required, globally for agriculture.
Well, how does the cycle work? Can you give it some detail?
Yeah. So normally the way nitrogen is fixed in the environment is through bacteria in the soil that are able to convert atmospheric nitrogen into fixed forms of nitrogen. Basically they can convert nitrogen into ammonia. And these bacteria are harbored by roots of certain types of plants, and mainly legume plants, where they create nodule-like structures. They look like a little tumor, but they grow on these roots, and they become the site at which atmospheric nitrogen is converted into ammonia, which is then a very easily assimilated form of nitrogen that can enter into all the biochemical processes, make all the 20 amino acids and the nucleic acids, and allow cells to function.
Is there any creature that fixes nitrogen out of the soil, or it's only out of the air?
There is fixed forms of nitrogen like nitrate and nitrite. So nitrate is a really stable form of nitrogen that you find in the soil. That would be the fossil nitrogen that's present in soil. And so that's certainly what's used by plants and microbes. And microbes can also convert or mineralize, atmospheric nitrogen into different sorts of microbes in the soil, can convert that into nitrate, which is then a fully oxidized form of nitrogen that sits in the soil.
But what's the preference and what's the mix? So ammonia is predominant, or is it various nitrates? Is it a whole cocktail?
Ammonia is a form that's used in all living organisms for the biochemical reaction. So that's the most reduced form of nitrogen that's used, in all the biochemical reactions.
Right, but the microbes that fix nitrogen, will they fix it to various different forms and use each of the forms? Some nitrates, some nitrites, some ammonia, etc., or do they predominantly only do ammonia?
So there are some forms that will make ammonia, and there are different types of microbes which will make different forms of nitrogen in the soil. So there are nitrifying bacteria and there are denitrifying bacteria, and they are present in the soil and they interconvert nitrogen into various forms. But as far as plants are concerned, they really need nitrate, as a fixed form of nitrogen that they can take up. And so that's why fertilizers tend to be, ammonium nitrate and that sort of fertilizer, which is used for plants to take up nitrogen.
Gotcha. So in a field with crops, is it possible to plant nitrogen-fixing cover crops, let's say, so you don't need fertilizer?
So that's a really good idea, and that's why we have these crop rotations, where people will be growing cereal crops and then have other crops that are legumes, which are cover crops. Then they or just alternate, between a cereal crop that needs nitrogen, fixed nitrogen, and legumes which can fix atmospheric nitrogen because of the association with these symbiotic bacteria, and then they are able to create, they have excess nitrogen that gets enriched in the soil, and so you have this nice cycle going. You can also have cover crops that are interspersed in tree crops and things like that, which can contribute to nitrogen in the soil. Apart from using manure and those type of things that also have fixed forms of nitrogen in the soil. So there are various ways to have nitrogen enriched in the soil through breakdown of plant material or urban refuge that can contribute to nitrogen. So there's a lot of nitrogen that gets into the environment from us, just excreting it out from our bodies as well as from our food sources into the environment. So there's a lot of nitrogen that's lost that seeps into the environment. Certainly, if you have the fixed forms like nitrate, they are more stable and they don't get into groundwater, whereas more assimilar forms of nitrogen like ammonia can get into groundwater, and then you have all sorts of eutrophication problems, as nitrogen seeps into soil because it's very highly toxic. Ammonia is very toxic.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now, back to the show.
So has anyone identified what are some of the, and it probably depends on what you're growing, but any idea of some of the ballpark ratios of the various, oxidized or reduced forms of nitrogen that are necessary?
It's certainly an area that most people farmers who grow are evaluating, how much nitrogen is there in the soil, and then they will put. But there's really no good way to know what the demand is for nitrogen and to give just that much nitrogen to plants so that they will just take up what they require and you don't have any excess in the soil. But unfortunately, since there's really no good sensor or mechanism to do that, most farmers are over fertilizing their plants, So that is the problem. It's not something I do research on. We are talking about a topic that I don't do research on, but I'm very familiar with since, tree nuts and wines require nitrogen, but they are more complex because they are long-lived, plants. They go through a cycle. So they are able to actually store nitrogen and recycle nitrogen. So they have a much more complicated way in which to utilize nitrogen. And therefore, it becomes even more complicated understanding, the demand of how to prescribe the right amount of nitrogen for an orchard or a vineyard.
Well, not only the right amount, but the right forms, right?
And the right forms, right? Yeah. So it's really, and when you grow plants in tissue culture, you need to provide them more organic forms of nitrogen, which are really very nice for them because they are able to use those organic forms of nitrogen in a much better way without having to have, things that form ammonia, which is more detrimental and more toxic to plants growing in culture. Because, I not only study plants, in orchards and vineyards, but we also study them growing in tissue culture. And as you know, tissue culture is going to become more and more important as we try to have plant material that we can eat. So then this is where the nitrogen is going to become a really important factor.
Gotcha. One question I have, we can switch to fruit trees and nuts and all that and wine. It's very interesting. Why does it take multiple years for a lot of fruit and nut trees to produce? Like, how does the plant know, this season, not yet, next season, yep, we're ready to go, let's produce?
So it's a really interesting question. And what happens with most trees, nuts, and wines is they have what you call a juvenile phase. So a juvenile phase is when they are growing vegetatively. That means they're producing just vegetative material, roots and leaves and branches and so on, right? But they are not producing any fruit, any reproductive organisms. And then they make a switch at some point where they become mature. And this time from juvenility to maturity is very variable with various different crops, tree crops. So when they become mature is when their vegetative buds will now develop, reproductive structures, flowers and pollen and ovaries that then, become the fruit that we eat, or the berries that we eat. So this transition from a vegetative bud to reproductive structure is very variable. And so, for example, in grape wines, it may take 2 to 5 years, depending on whether they will start fruiting. And for walnuts, that's the case too. It can take anywhere from 3 to 5 years before they start making fruit. Citruses are very variable. Some, it may take as much as 15 years before they start producing fruit. And so it's very variable among different tree crops and different varieties and different tree crops. Sometimes you can actually improve that or shorten the juvenile stage by using a rootstock. I don't know whether you're familiar that most trees, nuts, and vines are composite genetic systems where they have a root system that's different to the scion. So you have a rootstock that's grafted to a scion. The scion is the top part of the plant, which creates the fruit that we eat, right? But the rootstock is there to improve nitrogen assimilation.
Is the rootstock from the same plant or from a different kind of plant it's grafted on?
So it's usually a different species of plants in the same, genera as the plant. For example, in the case of walnuts, for example, top part of the plant is Juglans regia, which is the English walnut or Persian walnut. This is what we eat. But the rootstock is a variety of different Juglans species. It could be like, for example, in California, it could be Juglans heinzii, which is the California black walnut. It could be microcarpa, which is Juglans microcarpa, which is a Texas black walnut. It could be Juglans nigra, which is a eastern black walnut. So it could be a hybrid. And in most production systems these days use hybrids, where you have a hybrid between English, that means it's a cross between English and a black walnut. So you have a hybrid. That hybrid is known, in the case of walnuts, it's called paradox walnut. And it really stimulates the growth of the plant because it's a hybrid. It has hybrid vigor, and it makes the plant bigger and more productive. This is the case for all the rootstocks that are used to graft on peaches and nectarines and plums, prunes, apricots, and almonds also have a hybrid rootstock. In the case of grape wine, they also have hybrid rootstocks, which are native species of Vitis that are not related to Vitis vinifera. Vitis vinifera are the grape wines that make all the different varieties of wine. They are all Vitis vinifera species. But those all came from Europe. But in the United States, you have native species that were resistant to phylloxera, for example. As you know, phylloxera was a root insect that fed on roots that destroyed the entire wine industry in Europe, in the late 19th century, somewhere around between 1880 and that part, essentially destroyed the entire wine industry in Europe. And they had to move to using these rootstocks, American rootstocks, that were a hybrid species of different Vitis vinifera species that grow, in the eastern and southeastern United States.
How do you physically graft a root onto another plant? At what stage do you do this? How do they do it?
Yeah, this is a great question. So this is all done in nurseries. So that's why, in California, you have the nursery industry is like, I think it's worth farm gate value around $3 billion. And what they do is they provide plant material for growers to grow. All rootstocks are vegetatively propagated, as are the scion. And then when they are in a seedling stage, in a pot, they will graft them. They can graft them two ways. They can do what is called a bench graft, where they take the top and they make a cut and they splice them together and then hold them together with a rubber band. And what happens is that a vascular connection is made. You have a little callus that's formed, unorganized growth between the two pieces that are brought together. And then you have vascular connections that then establish, the xylem and phloem connections. And then you get a plant that you virtually can't see where the graft was done. You can also have what is known as bud grafting. So you have a plant and then you take a bud from the scion variety and then you bud it into the stem. You make a little cut a tea cut and then you slip the bud in there and then you let it heal. And what happens, the bud takes, then you cut the top of the other plant, the rootstock, and now this becomes the plant. And this is all done. There's a lot of art to it. Not everybody can do this effectively. A lot of home gardeners will do this. That's how you have a plant with, you can have a citrus rootstock with different citrus varieties, on the plant. You'll see that in many nurseries will provide you plants. You can have a prunus, which has apricots on one branch and cherries on another branch and plums on another branch or peaches on another branch. So you can, if you know how to do this, you can have a lot of fun grafting. There's a lot of pruning that's done and training. There's a lot of manicuring that is done on these plants, and it's the whole field of horticulture which allows you to do this. Every case, you must understand that these are all clones. So that's why Vitis vinifera may have a clone, a Zinfandel, for example, that you make Zinfandel wine out of. So all those plants are propagated from one plant. A good example is the Fuji apple, right? So the Fuji apple, all Fuji apples come from this one tree in Morioka, Japan, where they took branches or stems with buds on them, and then they grafted those buds to different rootstocks. And so you're propagating this one plant that sits in Morioka, Japan, all over the world. Everything is a clone. So they are clones because they'll have a name to it. So you have Fuji apple, Golden Delicious apple, this is a clone. Russet Burbank potato is a clone. Santa Rosa plum is a clone. Bing cherry is a clone. All these. But
if you have clones. If you have too many clones, I would guess that once certain pests, find them, that it could be incredibly detrimental if they attack.
That's right. Yes. So you could lose the clone, especially if it's very sensitive to a disease, which is something that we are trying to fix so that we can use, we can overcome the deficiencies in some of these and maintain quality, and productivity in a good way. But you're right. So some clones are quite resilient, but they are really valued for the quality that they give the consumer. And so in the case of wine grapes, you have Chardonnay, right, which is an important clone, and you can't make any changes to that because Chardonnay it gives you that particular quality of wine, or Golden Delicious apple. Some people like them and prefer them, yet others may like Granny Smith, And so
in a field, you could possibly help yourself by doing, I don't know if this is possible, but could you plant Golden Delicious and Granny and let's say 4 different kinds in a given field instead of just a monoculture? Would that help at all?
Yes, you could do that. It's really a question of then how do you harvest and segregate the different apples, but you certainly can do that. And most, backyard farmers will grow that, or small farmers would do that, have different varieties, and it gives them a production that spans, the entire season. This is true for peaches because you have early peaches and middle peaches and late peaches, something that come earlier in the season so that you can get a better price for them, and they may not taste as good as the ones that come later in the season, but yeah.
What's the expertise in grafting on various root systems? What do you need in a root system to help these trees grow? Can you plant? What are the characteristics of these root systems, and how was that discovered?
So the root systems are really environmental specific. So it depends on the type of soil growers have. They will choose a certain rootstock that will help them in that location. And so, for example, in grape wine in California, as you know, produces 80% of the wine in the United States, and they grow in these various regions. And if you look at these various regions, there are about, I would say, maybe 25 different rootstocks that are there. There are about 5 of them that are used most predominantly. And I've seen different growers will choose the rootstock based on the type of soil they have, the type of diseases that are present in the area. They will choose a rootstock. And then, so they've got to then, that's one choice. And then they've got to think what sort of top they want to have, what sort of wine they want to produce, whether it's a Cabernet Sauvignon. That's why you see in some areas you have really good Merlot growing, but, Cabernet Sauvignon doesn't do as well. So you've got to think not only of the root system, but you also have to think of the top part of the plant, how it would do in that environment, and, what sort of quality of wine it will make. By and large, grape wines are grown in most stressful environments because you get better quality of wine when the plant is stressed.
Let's go back to the life cycle, the vegetative versus the fruiting cycle of these trees. What factors determine, and can you force these trees to bear fruit earlier or faster than you normally would? Has that been studied?
Yeah, so there are lots of tricks. People try to take more mature tissue, from harvest from a plant. For the sign, they will take more mature branches, so they are further along in the developmental cycle, and then they will bud graft those onto rootstocks. There are some rootstocks that help plants mature faster. So you have that can happen for example, in apple, you have the malling series of rootstocks that can actually that can make the plant more precocious. That means it will flower much faster. So there are various tricks. There are ways in which you can prune and train plants that can help them mature faster. So horticulturists will use all sorts of tricks in trying to make the plants. They will bend the branches, try to create the structure of the plant so that it can mature faster. As you know, in grape wines, it's a really good process where they have to train the plant to have two cordons that are then sit on a scaffold, and then they have these shoots that come out, and then they train the shoots so that they will get into production faster. So a lot of the training and pruning systems are well established for different sorts of plants that and rootstocks and science that people know from a horticultural knowledge base, how to train these. But the environment plays a huge role. And so some environments, it may just take a longer time for the plant to get established, and so that becomes an issue.
So what is your research about specifically? What are you trying to figure out?
I work on trying to improve the productivity and quality of basically orchard and vineyard crops. And I study root systems a lot. We are trying to create roots that are more resilient to diseases. There's one disease, crown gall, that I've been focused on for the last many years, trying to find resistance to crown gall disease. This is a tumorous growth that many fruit trees and wines are very sensitive to, and we have come up with a solution where we can inhibit the formation of the tumor formation by this bacteria that is quite ubiquitous in agricultural soil. And so that's one of the things that we've been working on. We try to take a different perspective. I try to study the disease rather than the organism and its virulence, or the pathology of the organism. So I try to look at the plant perspective to the disease and processes, and try to understand how the pathogen is creating the disease, and try to find ways in which I can stop the disease from taking place. And so it's a little bit different perspective than a pathological perspective.
Well, what's an example of that? Can you go through one to make more sense?
Yeah. So in the case of the crown gall, which is a disease, it's a tumorous growth on the base of these plants, and it occurs due to wounding, natural wounds. These were the association between these tumors and these bacteria was established maybe 150 years ago. So we've known that, and we've studied the genetics of this organism now in great detail. So we know precisely how it is able to make these tumors, because it's a natural genetic engineer. It sends a piece of DNA into the plant tissues, which are in the wounded cells that are healing. It is able to send this piece of DNA into the nucleus, and encoded in this piece of DNA is several functions. And there are about 3 genes which we refer to as oncogenes. Oncogenes is borrowed from the cancer term, where there will be cancer-forming genes. The tumor is a form of cancer, but these are growth-enhancing genes that are expressed in the plant, and that's how you get the tumor. So what we've done is we've used epigenetic approaches. That means we control the expression. We put a system in the plant that downregulates the expression of these genes if it's infected. So in a way, it sort of works like a vaccination. You are producing something that basically downregulates the expression, and so you don't have the tumor formation. So the bacteria can still infect, but it just can't make the tumor because you put a system in that recognizes those tumor-forming genes or the expression of those tumor-forming genes, and essentially degrades the messenger RNA that's produced, that's required for that expression to create the tumor.
Well, what kind of off-target effects does it have?
It's so surprisingly accurate. It's sort of, this system, it's an epigenetic system that's present in all plants. It's referred to as RNA interference, and it's a system that's present in almost all eukaryotes have this system in them, and it's there to prevent them from getting infected by viruses. It's an antiviral system, and so essentially by giving them the trigger against these genes, it's an RNA-mediated process that is so accurate that it just takes out that messenger RNA. When you have a transformation by an incoming agrobacterium that's going to create the tumor, it essentially chops down just that RNA. It's extremely accurate.
Any off-target effects? I mean, do these plants grow and flower and fruit normally? And
yes, they just grow normally and fruit normally. They're indistinguishable. When we look at the siRNA that are formed, they're just for those gene targets that we've introduced. So it's a really very elegant system because it works at the RNA level, and it uses the size of the RNA is about 21 nucleotides long. So the possibility of making a mistake is infinitesimally small because, you can trace a gene by as much as 16 base pairs, right? So most PCR primers are in the 18 base pairs to 20 base pairs. So then you only will amplify that piece of DNA. So the accuracy is due to the base pair complementation that's taking place in the piece of RNA. And since that RNA is, 20 nucleotides long, it's accurate, and the accuracy is due to that. CRISPR systems also have a 20 base, region which they will recognize. But then there's a lot of interplay there so you have off-target effects in that.
Well, there must be some trade-off here. I mean, you're down-regulating certain genes. Those genes probably don't just have a single function. What else?
It's true. It's a good question. So it comes to what it means is that what is the homology between these genes that create these tumors, and there are 3 of them, and we knock out 2 of those genes. What is the homology between those sequences that you find in nature and plant genome sequences? That's really the question. If there was a similarity, and that similarity has to be in excess of 80% similar, then you would have a off-target effect. But we don't see that. There's no conservation. There's no similar sequence in plants. And so you have to avoid an off-target effect, you need to make sure that the similarity is less than 70%, and then you will not have the off-target effect. And if you want the system to work, you would have similarity in excess of 80%. And what is cool about this, as far as on the bacterial side, you've got to worry about bacteria having resistance to this mechanism. It turns out that almost all these genes are highly conserved among agrobacterium species that make tumors. They are there are more than 90% similarity between the same gene in all agrobacterium species.
Why are these tumors approximations of root nodules? Like what is the point of these tumors?
They are very different to root nodules. They are more like a tumor. They're more of a cancer. They are unorganized growth. So you'll see these big tumors.
So they're like a neoplasm, but they're okay. So
they're basically a neoplasm.
Are they similar at all in morphology to root nodules that are created on other plants that do harbor bacteria, that do harbor useful structures?
Yeah, they are different because the bacteria are not in the inside of these tumors. The bacteria sit on the outside of the tumors. So they are on the surface. Okay. In the case of nodules, the rhizobium are within that structure itself. And they need to be within that structure itself because you need to have less oxygen for nitrogen to be fixed. Here, what is happening is they are stealing nitrogen from the plant in the form of an opene. These are nitrogenous compounds that are stolen from the plant and exuded out to the surface where these bacteria sit, and they are able to metabolize these openes. So the tumor so the openes are these nitrogenous compounds that are also, the genes for their synthesis is also present on that tDNA, which goes inside the plant. Apart from the oncogenes, you have these opene synthesizing genes that are also present. And so the tumor becomes a factory to channel nitrogen, carbon and nitrogen from the plant to make this opene, which is essentially a conjugate between a carbon organic acid and a amino acid, basically. It conjugates an amino acid, it's an organic acid. And so the bacteria are able to metabolize it because they have these opene breakdown genes that can, and they are unique. Each agrobacterium, there may be about 40 different opene type structures among all agrobacterium species in the soil. And so they may have the different agrobacterium may make different openes, but they seem to use the same tumor-forming genes. Those are highly conserved. And they are mobilized horizontally. They are acquired horizontally among different populations. So as we have orchards that are, infected, there's no way you can avoid those genes being passed on to the other agrobacterium in the environment. So a tumor is not only a home for these bacteria to acquire nutrition, but they use it also to transmit those genes to different agrobacterium in the bacteria that can use those plasmids that contain this genetic material. It's passed down horizontally. So the problem tends to get worse. And so we think our solution is quite unique because we are stopping the tumor formation, so we are eliminating the home. So the bacteria don't have a home and a hotel to provide them the food source.
Okay, interesting. So, what other diseases are you trying to fight in these tree plants? And are you using the same methodology now that it works? You can expand to other types of.
Yeah. So we've been successful with the crown gall resistant plant in walnut, and we are sort of trying to look at this new regulatory process that APHIS has to see if we can have it released to farmers. We are now trying to make rootstocks. So we can take existing rootstocks that have all sorts of useful features and introduce these trigger genes so that will make them resistant to crown gall. And we're trying to do this in grape wine, 5 different rootstocks that we are currently trying to install the genes in. And then probably a rootstock for prunus. Prunus, as you know, is the family of the Rosaceae family that has almonds are part of that, and we grow so many almonds in California. The rootstock is very sensitive to crown gall, so we would like to make that resistant. And so that's on the crown gall side. We are also working on Pierce's disease, which is the disease of the top of the plant. So for the bottom part of the plant, for the rootstock, we think that RNAi approaches would be really useful. We could, in addition to crown gall, we are investigating how to make them resistant to Phytophthora using an RNAi strategy, and also to nematodes using an RNAi strategy. So that's the bottom part of the plant. For the top part of the plant, there are many diseases on the top of the plant, the cyan, the edible structure, where Pierce's disease is a good example. It's an insect-transmitted disease. I don't know whether you are familiar with Pierce's disease. It's a disease that showed up in California, in the late 19th century. They had an epidemic. Most of the wine production was in Santa Ana region, in Los Angeles. That was a hubbub of activity, and then was essentially destroyed completely because of Pierce's disease. Pierce's disease is endemic in the southeastern of the United States, where this bacteria is transmitted via insects. These insects infect the plant, and they are xylem-feeding insects. They are leafhoppers, and the bacteria sits in their mouth part and is able to migrate into the plant as it's feeding. And they sit in the xylem of the plant, which is the water element in the plant. And they are xylem-limited, very fastidious bacteria. And so we are trying to make proteins that will improve the immunity against these in the top of the plant. So we've engineered rootstocks that make proteins that will interfere with the growth and reproduction of these bacteria in the top of the plant. And we have been able to do field trials where the root system makes these proteins, right, secretes them into the xylem, and then they migrate past the graft union to the top part of the plant, and so they can protect the top part of the plant from coming down with the disease. So it's a trans-graft protection.
Okay, gotcha.
So this is what we are investigating, how to get resistance to Pierce's disease.
So I guess one thing that could be done is you could inoculate a graft or, alter it, up or down regulate the required genes. And then once that's done, then you graft it on, and now the new plant would have the properties you want.
Exactly.
And perhaps even biologically, you might not alter the plant that you're grafting onto. This was that's really cool.
Yeah. So this is a really cool thing. So because all these plants are vegetatively propagated, all you need is a good line that protects, And so and then once you have that, like we have that for the crown gall resistant walnut rootstock, it's a paradox walnut rootstock, really good. It performs beautifully in the field, protects against crown gall formation. Even though the crown gall bacteria are there, it can infect, but it doesn't make the tumor, right? So it's pretty cool. And then you can graft any variety on top of it, right? You could graft, all, different varieties of walnut.
That's interesting.
And they are not, certainly the bottom part, because the RNAi system is introduced via a transgene, so it is a transgenic rootstock, but the top part of the plant is not. And that would be the case with the grapevine too. The different rootstocks that we make that are resistant to Pierce's disease, the rootstock is engineered, whereas the top of the plant, you could put any. You could put Chardonnay, you could put Zinfandel, you could put Cabernet Sauvignon. You don't have to make any changes there, but you are protecting them against the disease through the root system.
Yeah, that's really cool. So what's the name for this process? Or have you created a name for it? I guess engineered rootstocks, or I don't know, disease-resistant rootstocks?
Yeah, I would say these would be disease-resistant rootstocks. And we hope to see if we can make them more resilient to climate also, the same way, by modifying the rootstocks in such a way that we could, make them more resilient. And I think this is where genomics is really helping us, the whole field of genomics, where we understand the genome, how it functions, and we can, it's because of genomics that we can target specific genes and pathogens, or we can enrich, we can help genes plant side, we can see why they become more sensitive, We can knock down the genes, for example, that's the case with the crown gall, right? We knock down the expression, and then we will get, more resilient plants.
Okay. Well, very good. Abhaya, what's the best way for people to find out more about your work? Where can they go?
I guess they can look at my Google Scholar profile, or if they can look at, I don't have a very extensive footprint where you can, but if you can see the publications I have in this field. If they just Google my name, I think you'll come across website and things like that.
Okay. I just wanted to make sure. Thank you so much for coming on the podcast. I really appreciate it.
Thank you so much for including me in your repertoire of people that you were getting podcasts from.
You're welcome.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.